Jeremy, if you could give $1 billion prize to the brain preservation or the connectomics or the dense reconstruction, the sectioning world, anyone you want, what would you give the $1 billion prize to? What's the problem that you have in reconstruction that you need to go away?
There is this sort of key challenge that is sort of still in its infancy of actually turning whatever we've taken from the tissue into a simulatable model. Until we've done that, we really have no real validation of the entire process. We want to be able to train a mouse or train a collection of mice in different behaviors, image them, reconstruct them, and simulate them, and observe these behaviors and sort of distinguish these mice. And sort of a challenge along those lines, I think, would really get to the heart of whether we've sort of made significant progress in this whole brain preservation.
What could happen that would really convince you that you had done a good reconstruction?
So as a concrete example, you could imagine you train a whole, some group of mice, each into separate complex behaviors that would ideally involve several of the senses and different types of planning and use of memory. And then after preserving these mouse brains and then reconstructing them and simulating them, you would want to be able to observe the same behaviors that you've trained. That really would indicate that you've gone a long way in being able to do this complete preservation and reanimation.
What Jeremy just laid out would be a fantastic experiment, but it is far beyond what we can do today. I think at one point it was reconstruct the connectome of a mouse. Okay, that is already far beyond what we can do today. There are things that we can do today that are not as complete. Let me give you one example. Tony Zadar, his group trained mice, I think it was mice, to lick in one direction if they heard a high tone and lick in a different direction if they heard a low tone. They could tell after the mouse was dead, and they sliced it, and they took a slice out that they did. It was electrophysiology, but what they were doing is they were counting synaptic connections into one area of the striatum versus a different area. And they could separate the mice into those that learned one direction versus those that learned a different direction. It's a trivial memory, but it shows, and it's one of several examples of showing that, yes, you can read off a learned behavior from connectivity. Have they done this with full animals? I think Robert has some other examples of C. elegans, perhaps.
Backing up one step here, what is the question, I guess?
I was asking Jeremy what would be useful for him in doing his job in trying to, and initially I thought he was going to tell me, I need better data, I need the knife to stop chattering when it's cutting, or like stop making strips. And he kind of turned it around on me and said that it's actually not the front end right now that's kind of limiting, but the fact that there's very little ability for him to validate whether his reconstruction is useful or not. He was actually throwing down the gauntlet for a pairing of behavior and reconstruction. We're all hopeful that the preservation will go well, that the reconstruction will go well, that once we understand the system, we could potentially have even like a sparse connectome or an error-contaminated brain sample that would still be salvageable. Help us understand, like, in connectomics, are people discovering like latent variables? Are there better ways to describe these densely connected kind of networks of neurons computationally?
I think it's reasonable to assume that if you lose one neuron, you're still going to be fine.
We lose 10,000. We lose 100,000 a day, by the way. Like, just while we've been talking, you're having a few die like every few seconds. And with them go about 10,000 synapses.
Half of the neurons, though, I think if you lost half of the neurons, there's a random half. I think probably there would be a lot of trouble with the reconstruction. Certainly at the level of individual synapses, there seems
the worst thing are, as you said earlier, correlated errors. So if you actually lost a random half of the neurons, you actually might not have the biggest problem. But if you lost like a number of neurons that was kind of like meaningful, like you only lost like a couple columns like from a particular cortical area, that would probably mean more than if you spread it out.
Well, in the fruit fly, you have cases where there's only 2 of a given neuron in the entire brain, and so certainly if you lost one of those, you're losing quite quite a bit. And so it's hard to say in humans. But yeah, I think if you say that you lost half of all neurons, whether that would, I think it's plausible that the reconstruction would be poor in that case.
I mean, aging alone, right? Like if you just look at the brain mass from when you're, 25 versus when you're 80, you're already in some cases losing half of your brain mass. It's amazing how much you can lose though with that process and still not like have it be catastrophic, right? Like if you deleted 10% of the transistors in a CPU, it would just fail to do anything. It wouldn't be decline. It would be like catastrophically ruined.
I think that we can look at a lot of different neural network models of biological systems, models of the hippocampus, et cetera, models of the visual cortex, and you would get real answers to that would make sense. For example, I read a paper recently where they literally chopped off with a laser microdissector in a live animal and a live mouse dendritic branches and recorded how the tuning properties of that visual neuron responded when they chopped off 30% of its inputs. And not much. So there's redundancy at all sorts of different levels. But I think we might be missing the point a little bit, because if I can ask Jeremy, if instead of missing half the synapses or missing half of the neurons, if instead you put only 1% of the volume, but you pulled that 1% of volume in random 1 micron cubes, which might happen from, I don't know, little ice crystals forming because of a bad preservation process, then would that, from a connectomics point of view, completely destroy the connectivity?
Right. Yeah. That sort of thing would definitely essentially disconnect all of the processes, and yeah, that would be catastrophic for the reconstruction.
And that's the kind of stuff that I think we should demand for anybody that might be offering a preservation to somebody. They need to at least be able to meet the brain preservation prize criteria.